Hello, my lovely anatomists and physiologists. Michele Glass here. We are finishing up our special senses chapter by looking at the special sense of vision. And this, of course, includes the structure and function of the eye. So we're going to spend a little bit of time here first going through some anatomy of the eye. So some of this is hopefully review of what you're doing in your lab work as well. And then from there, we'll get into the physiology. So we'll sort of be anatomists here for just a little bit of time. So I have some images here from our models in our lab. And let's start with this image where we have the intact eyeball. So when we think about the eyeball, we can think of sort of an onion, and it has three distinct layers, or those layers can be referred to as tunics. So the outermost tunic or layer is called the fibrous tunic, and that includes the sclera. The sclera is the white part of the eye, and that also includes the cornea. The sclera is the white part of the eye, really holding the eyeball structure in place. And then you have a transparent part that goes over top of where the light enters, and that is called the cornea. And so right here, you can maybe more easily see the cornea where it's been dissected. And you can see the cornea on this image as well. So the sclera is the outermost white part of the eye. It's opaque, it's fibrous, light doesn't penetrate. And then over this section of the eye where we would like light to enter, we have a structure called the cornea. If we remove the fibrous tunic, if we take off the sclera and the cornea, that's what we've done in this next image, and that allows us to see what's referred to as the vascular tunic or the vascular layer. And this is named because this is where you have a lot of vascularization, right? So that should make sense. So kind of covering the majority of the eyeball underneath the sclera, we have what's called the choroid. We have what's called the choroid. So the choroid is really this tissue that's providing blood vessels to the bulk of the eyeball. And then we also have what's called the iris. And the iris is this circular muscle that surrounds the pupil. And so the pupil is actually just this hole in the iris that allows light to enter. So the job of the iris is to control the diameter, the size of the pupil. So if your iris, it's a circular muscle, so if your iris is constricted, your pupil is very small and not a whole lot of light is entering. And if your iris is relaxed and that pupil is going to be large, it's dilated and a lot of light can enter. So in low light conditions, you would expect to have a dilated pupil, right? And so the iris is this muscle. It's the colored part of the eye. And then also we can consider the, I'm going to label it here as the ciliary body. We can consider this ciliary body to also be in this vascular tunic. And so the ciliary body is what's holding the lens in place, and it's helping to actually change the shape of the lens in order to help focus the light. So the ciliary body is involved in holding the lens and changing the shape of the lens to help focus the light. And what we're seeing in this diagram as well is we have this large kind of clear, transparent material at the back that's actually a fluid, and it's called the vitreous humor. So humor here is talking about fluid, and I use the word viscous to help me remember vitreous humor, Viscous means very thick, and the vitreous humor has a lot of thickness to it, a lot of structure. So the vitreous humor is important in helping to sort of maintain the shape of the eyeball, and it also helps to hold that retina in place. Okay, so far we have seen the outermost layer of the eyeball as the fibrous tunic with the white of the eye, the sclera. Attached to the sclera, you have what are called extraocular muscles. We studied several of those as part of our Anatomy and Physiology Part 1 lab. We have the cornea, which is that transparent covering over where the light enters. And then if we peel that away, we get to the vascular tunic, which includes the choroid, which is really providing a lot of the blood vessels to the tissue of the eyeball. We have this smooth muscle of the iris, which is the circular muscle controlling the diameter of the pupil. The pupil, remember, is just the hole that allows light to enter. We also have labeled the ciliary body, which is helping to hold the lens in place and will alter the shape of the lens. And so then the job of the lens is to focus the light. And we see also as we move into the back part of the eyeball, it's got this thick fluid called vitreous humor, really helping to maintain the shape of the eyeball, and we'll see also helping to hold the retina in place. Now, these are not, of course, all of the structures of the eyeball that we need to know. We're going more detailed in the laboratory portion, but this helps us understand our lecture conversation very well. Now, as we take a look at this particular view here, we are able to see the retina as the innermost layer of the eyeball. So this would be that innermost layer of that onion, right? This is going to be considered the neural tunic because this is going to contain our photoreceptors and this is going to contain our sensory neurons, right? We have this special part of the retina here, which is called the fovea. And the fovea has our highest, or we could say most accurate vision. This is because we have our highest concentration of what are called cone cells, but we also see that we don't have as much other tissue in the way to absorb those wavelengths of light. So maximum amount of light is coming in and stimulating these cells. We have our optic nerve, which is our cranial nerve number 2, coming out of the eyeball. And where that cranial nerve comes out, all you have is the sensory neuron fibers of those axons. You don't have any photoreceptors. And so we'll talk about that particular area where the blood vessels and the nerves are coming out of the eyeball as being called the blind spot. We're seeing a nice view here of the ciliary body. The lens has been removed. This little indentation here is the pupil. So what this picture is would be if you take the eyeball and you do a transverse cut. And so you're cutting it in half and you're peeling off the top half. And you're looking inside and you're seeing the retina and the fovea and the blind spot and where the optic nerve is coming out. Now, when we talk about our eyeball, we do have some important protections to the eyeball. Let me see if I can make that look a little nicer for you all. Some of you are thinking like, good luck, because it's always a little messy. And to you, I say, I know. So when we talk about some eye protections, we have, the eyelid itself is going to be an important protection to the eyeball. We do see that the eyelid is lined with what's called palpebral conjunctiva. So that's going to be the inside of the eyeball. And then over top of the eyeball part, we have what's called the ocular conjunctiva. Let me see if I have another name for that. It can also be called the bulbar. Bulbar. So the ocular conjunctiva is continuous with the palpebral. So the conjunctiva is this tissue that's lining the inside of the eyelid and then extending over top of the eyeball and then lining the inside of the lower eyelid. So the ocular or bulbar conjunctiva would be transparent so that light can penetrate and enter there. I also want to mention here, of course, your eyelashes. These are going to be important in preventing, large debris from entering the eyeball. And then also let's mention the lacrimal apparatus, and we'll go into more detail on that in your lecture, in your lab. So the lacrimal apparatus is the set of glands and passageways for tears. So of course, this is being produced at the superior lateral part of the eyeball, washing across and then draining through the nasolacrimal duct in the medial portion of the eye. When we talk about our retina, we're going to see that the retina contains our photoreceptors. So our photoreceptors are our special receptors that detect vision, and they come in two varieties. So we have either what's called a rod cell or a cone cell, and this has to do with the shape of the cells. When we talk about rod cells, we're going to see that they are going to be useful in low light conditions. They're often described as your black and white vision receptors. They're going to have stacks of membranous discs. They're going to contain the pigment rhodopsin. When we talk about cone cells, these are going to be excellent for your highlight conditions. So these are sort of like opposites. They're often described as involved in your color, and you have three different types. So you have a red cone cell, you have a green cone cell, and you have a blue cone cell. And when we say they're red, they're green, they're blue, that doesn't mean that's the color they look like in your retina. That means those are the wavelengths of light that stimulate these cells the best. Now, color gives you your most acuity of vision, your most accurate vision. So, and you can't see color without light. So it makes sense that your cone cell is your highlight conditions, that it's helping you to see color. Each of these different kind of cone cells, so whether they're responding to the red, the green, or the blue wavelengths of light, they have different forms of the pigment opsin. So pigments are special chemicals that absorb wavelengths of light, and in this case, when they absorb the wavelengths of light, they are stimulated, and that signal gets sent on to a sensory neuron. And rather than having stacks of membranous discs, we're going to have enfoldings of the cell membrane here. So the cell membrane is folding in to the cell rather than making discs. Okay, so let's try to do a little drawing of the retina. So the retina, excuse me. So the retina is just a few cells thick. So here we're going to look at the posterior part of the eye. And what we're going to see at the very back of the eye is we have a group of epithelial cells, and we talk about these cells as the pigmented epithelium. And again, if they're pigmented, that means they have those pigment molecules that can absorb wavelengths of light. So this is helping to capture extra wavelengths of light that are coming into the eyeball that haven't been captured by photoreceptors. You don't want those photons, those particles / wavelengths of light just bouncing around inside that eyeball. So the pigmented epithelium can absorb photons. So I'm drawing these, they look just sort of like a brick, right? So I'm just drawing like a brick. Okay, the next layer of cells that we have are going to be our photoreceptors. Now remember, we have two different types of photoreceptors. We have a rod and we have a cone cell. So I'm going to draw my rod cell. So I'm going to make it look sort of like a rod shape. And then I'm going to show my large cell body. And then I'm going to show an extension that has maybe like a little bit of microvilli at the bottom. And my rod has sort of embedded in that pigmented epithelium. So I'm going to call this my rod cell. And in that rod, I can do little kind of dashed lines to indicate like those are all my membranous discs. So that rod is made up of a stack of membranous discs. And then let's do a cone cell, and we're just going to draw one rod and one cone just to make our drawing easier. So now do sort of a triangular or rod shape at the top, and then do your enlarged cell body, and then do your extension with maybe like your microvilli at the bottom. Yeah, my drawings are amazing. So of course you'll want to take a look at your diagrams in your OpenStax book to help you with that, or maybe do a little Google image search. Now both your rod cell and your cone cell are going to be synapsing onto a sensory neuron. So now I'm going to draw a bipolar sensory neuron. So bipolar meaning, of course, I have my dendrite and then my cell body and then my axon. And these cells are actually described as bipolar cells. Now the bipolar cell is synapsing directly onto a neuron, and these neurons are going to make up here your optic nerve, which is cranial nerve number 2. So down here, this neuron is described as your retinal ganglion cell, and it's abbreviated as RGC in your textbook. So you have this lineage, or this, I shouldn't say lineage, but this line where you have your photoreceptor synapsing onto a bipolar cell synapsing onto the retinal ganglion cell. And the retinal ganglion cell makes up the fibers, the axons make up optic nerve, cranial nerve 2. So these are not the only cells that are here. We have also in between the photoreceptor and the bipolar cell, we have a cell that's called the horizontal cell. And then where the bipolar cell is synapsing onto the retinal ganglion cell, we have another neuron called an amacrine cell. And so what we see is the horizontal cells and the amacrine cells will modify the signal. So there's a little bit of processing that's happening at the retinal level. So in the retina, there's a little bit of processing that's happening by these cells. Okay, so we still need to talk about 3 more things for the eyes. It's a little bit more detailed. So I'm going to stop here and pick back up with the pathway in the next video. Stay tuned. As always, take care of yourselves and each other.